everyone. Welcome to Dr. Vanathi's Liquid Biopsy Podcast Literacy. Before we move on to the article review, let's touch some basics. This slide summarizes the type and lifespan of human cells. Globally, a small group of lab researchers have achieved a significant milestone by successfully culturing blood circulating tumor spheres and circulating cancer stem cells from a range of cancers including breast, gastric, colorectal, pancreatic, lung, brain and head and neck cancers. This pioneering work, as reviewed in Cancers by Us, has the potential to revolutionize cancer treatment. By identifying new CTC drug therapeutic targets, it could overcome the current challenges of conventional cancer treatment resistance. The technology is currently in the early stages of CTC drug testing following FDA standards and has the potential to significantly influence future Medonctherapeutics VP123 TGA patients' cell medicine treatment and predictions. Today, let's review a 2025 peer-reviewed article on pancreatic cancer circulating cancer stem cells published in Nature Portfolio Scientific Reports. This article, with a 3.8 impact factor, signifies its technical advancements, merits and high credibility in the field of oncology. In 2023, the diagnosis of pancreatic cancer was estimated to be more than 4,500 people in Australia. It is ranked the fourth most common cause of cancer deaths in 2022 with 3,902 deaths by 2024 as cited in the references link section. Globally in 2020, 4,995,773 new cases with 4,666,003 deaths were reported. In this article, the researchers were able to detect and quantify the number of tumor spheres as circulating cancer stem cells from 25 pancreatic cancer patient blood samples for personalized treatment. They successfully generated patient-derived xenografts using a chorioallantoic membrane CAM chicken egg yolk model. The number of tumor spheres correlated with tumor aggressiveness and UICC stages without complicated biopsy surgeries. These first findings paved the way for improved drug testing, treatment and diagnosis of pancreatic cancer, giving hope to medical oncology researchers and patients. Despite the project's limited funding, the good news is that the tests are now available to Australian patients. They can now submit samples in Perth with oncologist prescriptions, breaking down the geographical barriers and empowering patients. In the past, IP CTC technology has been extensively studied and commercialized, including the FDA-approved CellSearch system, iSet and StemTrack technology by Genostics in Australia with Germany. Now, can everyone afford it? Does Medicare cover it? Will it be accessible in all states and regional demographics? Coming to limitations of the study with critical evaluation. It's a 25 patient study without statistical significance and difference. Power statistics data required for clinical utility. Previous studies on breast cancer circulating tumor cells specificity on 72 early and metastatic cancer patients is different to pancreatic cells. Both the studies lack power statistical significance data. They cultured epithelial circulating tumor spheres. The technology is limited to IP CTC and its spheres. They did not detect EMT, MET, and all type of CTCs, including the FDA-approved cell search systems. How was the purity of the pancreatic circulating cancer spheres measured? Globally, how many clinical trials have been conducted on stem track technology to improve accuracy, sensitivity, and specificity? Organ specificity needs to be addressed. Pancreatic cell line positive spike test CTC data is required for measuring the stem track methods, accuracy, sensitivity, and specificity quantification. There is still an opportunity to rule out the patient's progenitor stem cell origin. These issues could be addressed more competently with available laboratory cell line spiking test resources. A good standard research laboratory practice requires cell line validation. The technology is immunostaining dependent. The staining may destroy and interfere with active cell surface proteins. The researchers could test on patient samples immediately because they could access samples in their country. These factors ensure the credibility and reliability of the research. This was made possible by the strict ethical approvals and high capacity for medical research funding in Perth. However, the research was not without its challenges, as indicated by the pre-terminated contracts due to limited funding or maybe competition disrupted the continuity of study and affected the pace of academic progress and advancement to the field. Despite these challenges, now the researcher commitment to our work remains unwavering with a new entrepreneurial spirit. To conclude this podcast with a happy note, I would like to leave you all with an open question. Why should we focus on patient-centered clinical trials for CTC drug development, reducing the use of animals and cost? Firstly, it mainly mimics the patient's current disease condition. Secondly, chicken, rodent, or monkey's blood vessel formation, angiogenesis model, genes, and immunity may vary from humans, which is the main cause for most drug testing failures. Thank you for watching. See you in the next podcast. Until then, stay happy and healthy.